I am Cassiano Welker from Universidade Federal de Uberlândia, Brazil, and I'm going to talk about updates on the phylogeny and classification of the tribe Andropogoneae. During this talk, I will focus on papers published over the last decade. The tribe Andropogoneae is a large clade of grasses that includes about 92 genera and more than 1,200 species. The tribe has a worldwide distribution and includes some of the most important plants for human consumption and animal feed, as well as for biofuel production. Some examples are sugarcane, maize, and sorghum. Therefore, Andropogoneae has a huge economic importance. As stated by Stepp and collaborators, the Andropogoneae feed and increasingly fuel the planet. All of us here probably don't go a single day without making use of these plants. So it's a very important plant from an economic point of view. But Andropogoneae also has a great ecological importance, since it dominates tropical grasslands around the world. As we can see here in some photos from Brazil, in this example, a grassland dominated by Saccharum intermedium. The morphology of the tribe Andropogoneae is very complex. Its members have paired spikelets, paired spikelets at each node of the rachis, normally one sessile and the other pedicellate. As we can see here in this scheme, the sessile spikelet and the pedicellate one. Here is the pedicel and the pedicellate spikelet itself. The same we can see here in the middle, the sessile spikelet with a long arm, the pedicellate spikelet, and the rachis internode. Each of these spikelets has two florets, and the lemmas are less consistent than the glumes. This is a big difference from tribes Panicoideae and Poaceae that Christian just discussed about, in which the glumes are generally membranous and less consistent than the upper antestion. Here, the glumes are the most consistent structure in the spikelet. In fact, the glumes are very important for the taxonomy of this group, and we use many characters from the glumes to distinguish genera and species in Andropogoneae, as we use, for example, many characters related to the type and form of the inflorescence for the taxonomy of this group. In Andropogoneae, the rachis disarticulation, the rachis generally disarticulates at maturity, and the spikelet pair generally acts as the dispersal unit. Again, as we can see here, the dispersal unit is generally formed by the sessile spikelet, the pedicellate spikelet, and the rachis internode. However, in some genera, the pedicellate spikelet falls first, and then the sessile spikelet is dispersed only with the pedicel of the pedicellate spikelet and the rachis internode. This difference is also useful to distinguish some groups of genera in Andropogoneae. So the morphology of Andropogoneae is very complicated, and the delimitation of genera and species is not easy. We do not have many qualitative characters that are used, and it's very common to differentiate species only based on quantitative characters. But not only morphology is complicated in Andropogoneae. Andropogoneae is also very complex from a phylogenetic point of view. The tribe is monophyletic, but the phylogenetic trees show short branches along the backbone of the trees, suggesting a rapid evolutionary radiation. In addition to that, polyploidy and reticulate evolution are very common in Andropogoneae, which makes the delimitation of genera species even more complicated. In this tree from Stepp and collaborators, each of these numbers and the black dot represent one independent allopolyploidization event that has occurred in the tribe. And I'm just showing here part tree. The phylogenetic trees of Andropogoneae show that Andropogoneae is formed by a core Andropogoneae clade that's formed by Andropogon, the type of the tribe, Schizachyrium, Botryochloa, and a few other genera. However, a large number of genera are placed outside of the core Andropogoneae clade, including Zia, Saccharum, Sorghum, and many, many others genera. During the last decades, the most comprehensive work on Andropogoneae published was this, published in Journal of Systematics and Evolution in 2020, which included a broad phylogenomic analysis of Andropogoneae and a biogeographic analysis, together with a new classification, a new subtribal classification for the tribe. This study was led by Michael McCain and me and supervised by Toby Kellogg, with input from some other colleagues. This study included the largest sample of genera to date, about 73% of Andropogoneae genera, and was based on 259 plastomes, 65 of them newly assembled for this study. Here we can see a portion of the phylogenetic tree of Andropogoneae, which shows that Andropogoneae diverged from Arundinelleae, its sister clade, in the early Miocene. We can see here also the map used for the biogeographic analysis, divided basically into continents, and we see that the biogeographic analysis indicated that Andropogoneae originated in East Asia, as most of the nodes of the backbone of the tree. Here is a summary of the biogeographical history of Andropogoneae. As I just said, Andropogoneae originated in East Asia. In this picture, arrows are proportional to the number of dispersal events that were estimated in the analysis. So we can see here a huge number of dispersal events from East Asia to Africa, to Oceania, and to West Asia. A smaller number, but still multiple independent events from the Old World to the New World, first to South America, and then to Central and North America. This analysis indicated about 300 independent dispersal events that have occurred in the evolution of Andropogoneae. In fact, the morphology of the diasporas of Andropogoneae, with a lot of trichomes in most of the groups, are consistent with dispersal by wind. Based on the plastome phylogeny and also using nuclear sequences, nuclear phylogenies from previous study, we could reorganize subtribal classification of Andropogoneae. Two new subtribes were proposed in this paper, Hitacnini and Chrysopogonini, and most of the other subtribes were recircumscribed. Some of the genera from Andropogoneae were placed in set sets in this analysis, either because they did not have a strong support in our analysis, or, more frequently, because the phylogenetic position in the plastome phylogeny was different from the phylogenetic position from nuclear studies published before. In this case, we decided to keep this genera in set sets until we have further evidence to correctly put them into the subtribes. A few genera in Andropogoneae were also placed in set sets because they have never been included in any molecular phylogeny so far. This is the case, for example, of some monotypic genera from India that we are now taking efforts to try to sequence these plants for the first time. Here we can see some more clades of Andropogoneae accepted as distinct subtribes. This new classification was therefore based on our molecular data, both plastomes and nuclear data. Previous classifications were based mainly on morphology, resulting that most of the subtribes, except until then, were not monophyletic. In this new classification, we accepted 14 subtribes, all of them monophyletic, and 92 genera. About 90% of the species could be assigned to a subtribe. Here we can see a summary of the phylogenetic relationships of these subtribes. In bold, we can see the names of the subtribes and also the insert set genera that need further studies. We can also see here, in parentheses, the number of the genera and species of each lineage. This broad phylogenomic analysis also indicated that several Andropogoneae genera are not monophyletic, including some large genera of the tribe, such as Saccharum, Andropogone, and Schizachyrium. Some of these genera had already been indicated as non-monophyletic in previous analysis, as is the case of the genus Saccharum. Saccharum, in a broad sense, includes about 40 species, but some authors divide the group into two genera, except the genus Eriochrysis as a distinct genus. In this circumscription, the genus Saccharum, in the strict sense, would be restricted, would include only Old World species and Eriochrysis Old and New World species. The main morphological difference to distinguish these groups would be the absence of an in the spikelets of Saccharum, in the strict sense, and the presence of annate, or at least mucronate, spikelets in Eriochrysis. Some other authors still divide Eriochrysis in two sections. Eriochrysis section, Eriochrysis, including all species from the New World, and Eriochrysis section Rhipidium, including the Old World species. Some authors still consider that the Old World species of Eriochrysis would be a distinct genus, the genus Rhipidium. The circumscription of other groups within the Saccharum complex is also unclear, such as Narenga and Miscantidium. Our previous phylogenetic analysis focused on South American taxa of this group, demonstrated the allopolyploid origin of the Saccharum complex. This study was based on several low-copy nuclear genes. So each polyploid species in this analysis have two or more terminals in the tree, corresponding to the different paralogs of the nuclear regions. So we can see here the two clear genomes of the Saccharum complex, which we arbitrarily called genome A and B. And this analysis also showed us that Rhipidium belongs to a distinct lineage from Saccharum. Although the morphology of this group is very similar, Rhipidium belongs to a distant lineage from the remaining members of the Saccharum complex. After that, we investigated this based on plastome sequences. Some phylogenomic analyses were performed, and we obtained the same topology regarding the position of Rhipidium. So Rhipidium forms a distinct lineage from the Saccharum complex. The circumscription within the Saccharum complex is still unclear, especially regarding the genus Eriochrysis. If we consider this as distinct genera, the presence or absence of ans and the number of anthers would be morphological characters that would separate these genera. We are now conducting a broad phylogenomic analysis of this group, sequencing many species of all these putative genera of this group to better investigate the circumscription in this clade, in this complex. Morphometric analysis performed with the Saccharum complex also showed many morphological difference, strong differentiation between groups of specimens and species in this complex, corresponding to the distinct putative genera in this complex. As we can see here, Saccharum in a strict sense, Narenga, Miscantidium, Tripidium, and Erianthus. Surprisingly, we also found differences between North American and South American species of Saccharum. One of my graduate students is now studying leaf micromorphology and anatomy of this group, and she has found some interesting anatomical differentiation between North and South American species of Saccharum. So they may consist of two distinct lineages that we need to investigate further in next studies. Moving forward, Andropogon is another very complicated genus within Andropogoneae. It is the largest genus of the tribe. It includes more than 100 species, and it is clearly polyphyletic. These species are placed in several clades along the phylogenetic tree, and many modifications will be required to make Andropogon monophyletic. Last year, Maria Vorontsova and colleagues published a paper in which they expanded the genus Anaterum to include many species of Andropogon, many of them from South America, for example, including Brazil. So this was the first step in the to circumscribe Andropogon, accepting Anaterum as a distinct genus with many of these species of Andropogon. But the remaining species still form many independent clades that will need to be further investigated. The main challenge here is to find the morphological characters that correspond to these distinct clades, distinct lineages of Andropogon. And a last example, the genus Schizachyrium is also very complex from the phylogenetic point of view. Recent studies have shown that the genus is not monophyletic and is formed by at least 3 distinct lineages. The main lineage, which includes the type species Schizachyrium condensatum, can be distinguished by the reduced spikelets without lemma and pallia, as we can see here, the sessile spikelet in the middle, and here the pedicellate spikelet, very reduced in this group of species, and the lower glume of the pedicellate spikelet with an an that varies in size. Other species of this genera generally have both spikelets developed. Sometimes the pedicellate spikelet is as big as the sessile spikelet, and the lower glume does not have an. Again, we need more studies here to make Schizachyrium monophyletic, and we will probably need to describe new genera to make Schizachyrium monophyletic. In addition to problems in generic delimitation of genera in Andropogoneae, we also have many problems of species complex in this tribe. I will show just one example here, which is the Schizachyrium condensatum complex. This species, which is the type of the genus, is characterized by a congested inflorescence, which is the main morphological character to distinguish it from Schizachyrium microstachyon, which has a lax and open inflorescence. The plants are completely different in the field. However, we do find many intermediate morphologies between these two extremes, from a congested inflorescence to an open inflorescence. These intermediate morphologies were described and accepted by some authors as distinct species. Some of them are listed here. Some authors consider all this group as a single species. In this case, Schizachyrium condensatum. Other authors consider and accept distinct species in this group, as we can see the name of some of them here. We are performing now phylogenetic analysis based on multiple low-copy nuclear genes to investigate species delimitation in this group. We are using low-copy nuclear genes and multiple individuals of these putative species. And what we have seen, we are still adding sequences to this analysis, but we have seen that multiple individuals of each of these putative species, identified by morphology, form a clade, indicating that we do not have a single species in this group, but we do have many different lineages, many different species in this complex, correlated with the morphology and the traditional taxonomy that we have. As the conclusion of this talk, we can say that many more changes are expected in Andropogoneae in the following years, especially concerning Andropogon and Schizachyrium. We will probably see many more genera being described to make this big genera, this large genera, monophyletic. Thank you for your attention, and I'm happy to answer any questions.
Thank you very much. Questions? You mentioned, well, first of all, I really enjoyed your talk.
Thank you.
But also, you mentioned that the spikelets of Andropogoneae often have those long awns, and it seems to be associated with wind dispersal. I've noticed when I've been working in the field that they also really easily get stuck on my clothes.
Yes.
And I'm wondering if there's any evidence that some of these dispersal events, these long-distance dispersal events, might have to do with bird migration, for example.
Yes, the ants are very interesting. They seem to have several functions in Andropogoneae. I have a graduate student studying a PhD now that will focus on the function of ants in this species. And it seems that it's useful to make the seed in the soil to germinate for dispersion. So it's a very interesting structure in these plants.
I'm really curious to see what he finds out. Thank you.
Thank you.
Another question?
Thank you.